This is Marcus. We have followed him for a while now and have seen how he eats, runs, and works. All of these activities are operated by his brain, the control center of the body. If we take a closer look at the brain, we can see that it mainly consists of nerve cells, such called neurons. If you look at the neuron in the brain, you recognize that each is connected to another. A neuron of the nervous system consists of a cell body called the soma, an axon, and a synaptic terminal. The synaptic terminal is connected to the dendrites of the second neuron. Every signal moves from the dendrites of the soma along the axon to the synaptic terminal over to the next neuron. The site fulfilling this job is referred to as the synapse. But how exactly is this signal, also known as the action potential, forwarded to the next neuron? When an action potential reaches the synaptic knob, the membrane is depolarized and voltage-gated sodium channels open, causing an influx of Na+ ions. At this point, voltage-gated calcium channels on the presynaptic membrane are still closed. The change of charge inside of the synaptic knob causes the voltage-gated calcium channels to open and let calcium 2+ ions enter the presynaptic neuron. Inside the presynaptic knob, vesicles containing neurotransmitters are present. In this case, the neurotransmitter is acetylcholine. The presence of calcium 2+ ions in the presynaptic neuron causes the vesicles to move towards the membrane and fuse with it, releasing acetylcholine into the synaptic cleft. This process is also very well known as exocytosis. The neurotransmitters that have been released into the synaptic cleft function as receptor molecules for ligand-gated sodium channels on the postsynaptic membrane. When the neurotransmitter binds to the receptor site, the channels open and Na+ ions present in the synaptic cleft move into the postsynaptic neuron, depolarizing it. This is where the action potential is transferred from one neuron to another. This is also referred to as excitatory postsynaptic potential. Eventually, the signal transmission should end. Therefore, the neurotransmitters left in the synaptic cleft have to be degraded by enzymes. These enzymes are in this case also known as acetylcholinesterase. With no more neurotransmitters in the synaptic cleft, the ligand-gated channels close and no further depolarization of the postsynaptic neuron can take place. Acetylcholinesterase cleaves the neurotransmitter into acetate and choline. These products are then again taken up, for example, via an Na+ choline symporter. There, both products are recycled and reused for the formation of new neurotransmitters. This process can be depicted in a curve as such. First, the synaptic terminal is at rest with a membrane potential of -70 mV. When an action potential arrives, the voltage rises. This is when the presynaptic knob is depolarized. The voltage falls and the presynaptic knob repolarizes when the action potential is transmitted to the postsynaptic neuron and it returns back to its resting stage.